Right, this is a question looking at a practical that was done looking at the percentage of stomata and number of stomata on the underside of a leaf. Right, remember that we always look for the number of stomata on the underside of the leaf because on the upper side there's very few. If stomata were on the upper side of the leaf, the rate of transpiration would be too high and the plant would lose all of its water. Right, in this sort of question, they've started off by giving you a method. It's really important that you spend some time reading that method properly, maybe go through, highlight keywords and such forth. You will need to be using the method in the answers. So the method follows that you peel off the lower epidermis of the leaf, you cut the epidermis to equal sized pieces. Right, remember that cutting it to equal sized pieces will be your control variable. Place each piece of the lower epidermis into a different petri dish. Add 5 cm cube of salt solution to each petri dish. Each petri dish should have a different concentration of salt. The 5 cm cube, so the volume that you're adding, is also a control variable. However, changing the concentration will be your independent variable because it's what you're changing. After 1 hour, view each piece of epidermis under a microscope, 4 × 400 magnification, and count the total number of stomata open. So the number of stomata open will be your dependent variable. Right, so the first part of the question is asking you to calculate the value of x in table 2. Right, all you're doing is working out a simple percentage. So remember, it's only the number of open stomata divided by the total number times by 100. So in this case, 6 divided by 7 times by 100, which would give you 85.7%. In this case, because you're talking about the number of stomata, you can't have half the stomata, so I would round that up. Part 2 goes and talks about one conclusion that you could make from the results in table 2. So basically you just need to say what you're saying. So the lower the salt concentration, the greater the number of stomata that were open. You will need to refer to both the independent and dependent variables when answering that type of question. Part 3, how could the students find out what the concentration of salt would be if the result of half the stomata were open? Easy way to do that. On a question like that, always talk about plotting the results on a graph, then going and reading off the result off the graph. Right? Sometimes you might need to describe it in more detail, sometimes you might need to go and draw the graph. In this case, because it's only worth 1 mark, you just need to make some reference to the fact that you will need to draw a graph to get that information. Part 4 goes, so a student measured the real diameter of the field view to be 0.375 millimeters. Calculate the number of open stomata of the leaf epidermis placed in a 0.4 molar decimeter cube salt solution. Right, this is where they've not given you the equation to work out the area of that circle of epidermis. So you would have to remember that to work out the area of a circle, you're going to have to use pi r squared. So simply, you take, well, if you had a normal calculator, you'd start off with 0.375 times by 0.375. Always remember to press equals. In between doing anything on calculators, make sure you're pressing equals, or else you run the risk of getting the wrong answer. Then times that by pi, which is 3.14, which equals 0.44. Then all you're going to go back to is the table on the previous page, look up 0.4, so the percentage of stomata that were open was 40%, so the number of stomata that were open was 4, so all you're going to do is times that by the number of stomata that was open. So that'd give you 1.76, so you could go and round that up to 2. So the next part of the question says that figure 6 shows guard cells surrounding a closed stomata and 2 guard cells surrounding an open stomata. Now it gives you a little bit of information on this. When light intensity is high, potassium ions are moved into the guard cells. Describe how the movement of potassium ions causes guard cells to open up. Right, this is something where people lose easy marks from because you overcomplicate it. People think, oh, I know nothing about the movement of potassium ions, I've never looked at that before. All it's talking about is the fact that if you're moving potassium ions in, you're going to lower the water concentration inside of the guard cells, therefore more water would move in. If you look at the diagrams, the guard cells surrounding the open stomata are a lot bigger because they've absorbed more water. As stomata absorb more water, they go and bend and it opens up. So, to answer this question, the movement of potassium ions would lower the water concentration within the guard cells. Therefore, more water would move in, there's your second mark. By osmosis, there's your third mark. The fourth mark would come from as more water is forced into the guard cells, it causes them to bend open, or bend, which opens the stomata. Right, go back, have a go at this question in your specimen paper. Any parts of the answer that you're still unsure about, go back and watch again.